Hello, my name is Lo Clark, and I'm going to be doing the demonstration in this video for the streak plate for isolation. Only a couple materials we'll need for this one. We're going to need our Bunsen burner. I've got a nutrient agar plate right here. I know it looks slanted, but we're going to use this as our tube of broth. Our inoculant loop tool right here. And then to demonstrate, I will replace the tool with a pen to show the streaking. We're going to need some safety material. We're going to need goggles for our eyes, gloves for our hands, and we'll need like a lab coat or apron to protect our body. To begin, we're going to start by sterilizing our inoculant loop. It's this guy right here. I'll run that guy through the flame and sterilize him. I'm going to tilt this down so that you can see a little better the rest of the demonstration. We're going to take our broth, we're going to remove the cap, we're going to take the tip of that broth and we're going to run it through the flame. That's going to sterilize the tip. It's also going to remove any possible air that's inside of it. Very gently, I'm going to take my inoculant tube, I'm going to dip it once into that broth. That's going to grab the bacteria, run that tip right back through the flame once more, and put the cap back on. We've got our media on our inoculant loop. I'm going to replace this with a pen to show for streaking. So this is now our inoculant loop. Starting in quadrant 1, very gently I'm going to take that loop and I'm going to streak it into that quadrant. Just like so. So we will have our first streak, that's our quadrant 1. We're going to take the inoculant loop, we're going to run that back through that Bunsen burner flame, turn our plate. This is going to be our second quadrant streak. It's going to come from the first quadrant, pull just from the edges, nice and light streaking. So as you can see, it will cross through that first quadrant. Sterilizing our inoculant loop again, turning the plate another time. Our third quadrant, like the last one, we're going to take it and cross it through that second quadrant when we're doing our streaking. So there's our third quadrant, crossing through our second. Re-sterilizing our loop once more. Giving that one final turn. I'm going to cross through that third quadrant into the fourth, and we're just going to lightly drag that down the center. I'm going to go ahead and sterilize this for a final time. We are done with our inoculant loop tool. Our final streak is going to look somewhat like this, coming down the middle. It's crossing through just that third quadrant coming down. Now, in order to finish this, we need to place the cap back on. We're going to incubate our inoculated media for about 24 to 48 hours in a warm environment. And that's going to allow that bacteria to grow. And once it's done growing, you'll start to see potential colonies growing. And they're going to look something similar to this, where you will have like single dots, and that's going to be a single unit colony right there. Not going to be the whole streak, it's just going to be these little dots that appear. And that right there is our streaking plate for isolation demonstration.